I can start getting everybody's attention up here, eyes and ears. So on Friday, we started talking about the process of transcription. Transcription means to make a copy. And so we're making a copy of DNA in the form of RNA. Yep. And when we look at a gene, which is the unit of DNA that we're transcribing, right, that we're going to end up making an RNA molecule from or a protein from, there's 4 important parts we discussed towards the end of class. The first one we talked about was this blue one here, which is called the promoter. That's just the start of the gene, right? So we have the TATA box here, which is going to help to recruit some of the important proteins we need to get the process started, right? Then we also have this red area way up here, which is called the enhancer. Do you remember what binds to the enhancer? Transcription factors. So these are proteins that play a factor in transcription, yeah, and binding stuff, yeah. And so that's either going to allow us to make more copies of the gene or less copies of the gene. That's part of the control portion of this, because there's 20,000 roughly genes in your genome. Does every single cell want to be making all of those proteins all the time? No. Okay, so you have 215 different cells. They only make a certain amount of those proteins at any point in time. Okay. Otherwise, you'd be wasting lots of energy, and your cells would get all gunked up, right? You'd have proteins everywhere, and nothing would be able to move around and get anything done. Okay, so that enhancer is really important for regulating when a gene gets copied and how often it gets copied. Okay. These other 2 parts here, we'll spend more time on today. The green sections are called exons. That is the code that's actually going to get used, right, to make our protein. We also have these sections in between called introns, and we'll talk about this more, but they're going to actually end up getting cut out of the code. So we're not 100% sure all the roles those introns have, but when we talk about them a little bit more today, you can probably get some ideas on why they might be there. So this lecture, we're going to go through the steps in a little bit more detail of how this happens. And then we're going to have some discussion questions to look at. So up here on the board are a couple of kind of more oversimplified views. So this is probably similar to something you would see in a more basic textbook. This is from Khan Academy, like a very basic overview of replication. And so in this case, it looks like here's our blue DNA molecule. What do you think this purple thing here is supposed to be? The mRNA. And notice there's light purple and dark purple sections. What eventually happens there? It looks like the purple sections get kind of cut out. Those are the introns that are getting cut out. And we end up with our mRNA molecule. Okay, and then it leaves and it gets translated. Here's another kind of, so here's the mRNA molecule and here's the introns that are getting spliced out. Okay. So transcription is definitely a multi-step process. So we're going to look at each one of those steps a little more detail here. And again, this is very heavily regulated because we only want to make certain proteins at certain times in certain types of cells. So there's about 100 proteins that play a role every time we want to make a molecule of RNA. Okay, so lots of proteins are involved in this. So that's why we have both the promoter and the enhancer regions for those things to connect to. Okay, so a lot of those are different transcription factors. And of course, we have our main enzyme we mentioned on Thursday, which is RNA polymerase, right? Enzyme that makes RNA polymers, puts RNA nucleotides together. And so this is going to also be a very, again, highly coordinated. So things have to fit together all in exactly the right way for everything to work the way it's supposed to. And then off to the side here, this is just showing the quaternary structure of RNA polymerase. It's a huge protein. And so there, all these different colored blobs here are all different polypeptides that have joined together in that last quaternary structure, right? So it's a huge enzyme. It both can separate the strands and add nucleotides at the same time. So really, really big protein, huge enzyme. So step number 1. Yeah, feel free to come. Nobody else is there. Formation of the pre-initiation complex. Sounds super fancy. Formation, you know, we're forming. What do you think pre-initiation means? Before starting, and then complex. So we're gonna have a bunch of proteins that are all joining together here on the strand. And so we're going to be looking at, sorry, this is kind of a little bit low to the screen, hopefully everybody can see it. We're going to be looking at this diagram here, and it's going to be changing as we go through this process. And in your group, you're going to be talking about what you see happening here. Okay. So first of all, we have our gene, right? What do you think this is supposed to represent? Can you see what it's labeled? TF. Yeah, what do you think TF stands for? Transcription factor. Okay. So we have a certain transcription factor that starts to bind to the promoter, right? So the first transcription factor binds to the promoter. So I'm just going to show you the picture, but you should probably put something down in your notes for what you see. Okay, so make sure you're writing down what you're seeing in your notes. Okay. What's happened now? There's some more transcription factors. So even more transcription factors bond to the promoter. You notice they're actually attached to this first one, right? So we're adding transcription factors on, more transcription factors. And then finally, what attaches to those? RNA polymerase. Once we get certain transcription factors attached to the promoter, then RNA polymerase can bind. And I'm not sure what happened. So I think this closed word got cut off on the very first slide. So this is also what we call the closed pre-initiation complex. So this has to happen before transcription can occur. And the word closed refers to the fact that our DNA strands are still stuck together. So the DNA strand is still closed, it hasn't opened yet.
What's the difference between like the first, second, and third types of RNA?
They transcribe different types of RNA. So RNA polymerase 2 is the one that transcribes mRNA. So you don't have to know those. You have to be able to match those. So yeah. So this is a gene that's going to code for a protein. But if it was a gene that was going to code for like a tRNA molecule, you'd have different transcription factors and different RNA. Okay. So hopefully in your summary there that you wrote as you went along, you say something along the lines of the fact that multiple transcription factors and RNA polymerase have to join at the promoter, right? So that's kind of step one. We're just gathering all these proteins in the right place. Lots of proteins involved in it's very highly regulated process. We only want to make certain genes at certain times and certain cells. Step number 2, we're going to open the pre-initiation complex. So, if you take a look down there, go ahead and talk to your group members. What do you notice has happened? Don't make it too complicated. Well, what's happened here? We started to split apart the DNA strands. Okay, that's all that's happened here. Is that we've split the DNA strands apart. We're starting to unzip the DNA. Okay, so now we're having closed DNA, where both of the sides are stuck together, to open DNA. So now we can get in between those strands. We do also have this. You can't really see this here, but there's more transcription factors that bind. And we're opening up the molecule, right? We're splitting apart the DNA. Remember, RNA polymerase is only going to copy one of the strands, not both of them. So whatever strand is getting passed here to RNA polymerase, that's the strand that's going to get copied. So we get all the right proteins there. Now we're opening up the molecule. We pass the strand that's going to get copied to our RNA polymerase. What do you think RNA polymerase is going to start doing now? Start the transcription process. Yeah, it's going to start adding complementary base pairs of RNA to that strand, right? Before it does that, though, we actually have to do some modifications to the RNA polymerase itself to get it started. What do you think P stands for?
Phosphate.
Phosphate groups. Do you remember what charge phosphate groups have? Negative. So when you attach negative phosphate groups to a protein, those negative charges are going to interact with all the R groups around it, really pulling on your DNA structure knowledge here. And so when you add phosphates, that's going to change the structure of the enzyme, and when you change the structure, you're going to change the function, right? So these phosphate groups, when we modify that polymerase, that's going to cause the protein to change shape and allow it to start copying the DNA. So you'll see, we'll talk more about these phosphate groups when we get to cell division, but anytime you add a phosphate group to a protein, again, you're going to change the shape. We add phosphate groups to proteins a lot. Oh, sorry. We need to go back. Our RNA polymerase has started to copy our DNA. So this purple molecule here is going to be our new mRNA molecule. So it's going to continue to move along the strand. The summary here also notes that there's a cap, a protective cap on the 5' end of the molecule. Remember, we're copying from 5' to 3', building from 5' to 3' is like we do when we copy DNA. And so that protective cap is going to make sure that the molecule doesn't start getting cut apart. What
was step 3?
Step 3 was we added these phosphate groups onto the polymerase and started. In the quick check, there's like a question where you have to order the steps. They're not worded exactly like this, and that's kind of on purpose. So you have to think about what's happening here and not just memorize the words. So when you get to that part of the assignment, you kind of keep that in mind. But in essence, we started copying our genes. We put this protective cap on our molecule to keep it from getting broken down or damaged before we can use it. Am I going through this too fast? So step 5, this is the major step. Like this is when all the copying is happening. So elongation just means we're building onto the strand, right? We're making the mRNA molecule longer. This is also when we do our splicing. So when we cut out our introns and put the exons together, and so take a look at the diagram here. Go ahead and talk in your group for a couple seconds. What do you notice is here that wasn't there on that slide? Yeah. So I want to point out here, elongation factors. These are proteins that play a role in elongation. We have splicing factors here. These are enzymes that play a role in splicing. Okay, so all of this stuff is happening at exactly the same time. So again, very highly coordinated. So as we are copying that DNA, as we're making our mRNA molecule, we have all these other proteins that are aiding in that process, including these splicing factors that are cutting out the introns. Okay, so they're removing the introns. If you look at where this is lining up on our gene, right, on either side of our intron, we're cutting it and putting the ends back together. Okay, so we're splicing and cutting out those introns. Yeah, you might have noticed our phosphate groups have moved from one location to other locations on the polymerase. That's not anything that's going to get tested on or whatever. Just might be something you noticed. So at the end, the DNA or the RNA polymerase is going to hit this stretch of DNA that's just T, like sometimes like 50 to a couple hundred thymine bases, which means what RNA base is this going to be? A, like sometimes that's like 250 A's in a row. We call this the polyadenylation site. What does poly mean? Many. Adenyl, adenine site. So we add a ton of adenines onto our RNA molecule, and that tells all the other proteins that this is the end. Okay. So it's like hitting the end of the book, right? It's like the end. Plop. Okay, so we're done copying our RNA molecule. And so at this point, there's another enzyme that's going to come in and cut our RNA molecule. So when that RNA polymerase crosses that site, when it makes all those A's, we call that region of the RNA molecule the poly-A tail. Again, poly just means a lot of A's. And at that point, our transcription is complete. So we have our 5' cap. We've spliced out the introns. We have a poly-A tail. That poly-A tail actually also gives some protection to the RNA molecule, so it doesn't get like cut to pieces right away. But you might notice that RNA polymerase doesn't actually know it to stop, so the RNA polymerase is going to keep going. And so the next couple of slides are more just for fun than for anything else. But so at that point, we have our RNA molecule. It can then leave the nucleus and go on to translation, which we'll start talking about tomorrow. So tomorrow, I'm going to have a discussion on Canvas. We'll have a video, and then you'll have to respond to some prompts to get credit for that. So that's what you're going to do tomorrow while we're out. I can't do a Google Meet because we're supposed to be like helping with iStep the whole day. I think it's kind of dumb, but anyways. So RNA polymerase doesn't know to stop. And you can just kind of watch this. You don't have to have this memorized, but there's a little enzyme called exonuclease that moves along. Oh, we're on the wrong way. There's our Pac-Man exonuclease. He moves faster than polymerase 2, so eventually he's going to catch up with polymerase 2, and he knocks it off of the DNA strand, and then RNA polymerase. I'll call it the torpedo model. So
this is not school appropriate.
Okay. So we watched this version, this series for replication. They also have one a simple video on transcription. So what do you think this red twisty molecule is? DNA. What do you think this big blue, huge blue blob protein is?
RNA polymerase.
RNA polymerase. What do you think this yellow thing being made is?
RNA.
The mRNA, yeah. So we'll go ahead and watch this real quick.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene.
Okay, so what are these right here? Specifically, transcription factors, they're bonded to the promoter, the promoter, right, of the gene.
A gene is simply a length of DNA instructions stretching away to the left. The assembled factors.
What's that?
Oh, that's the
That's RNA polymerase. Yeah.
There's triggered the first phase of the process.
There's some other transcription
Reading out the
factors.
information that will be needed to make the protein. Everything is ready to roll. 3, 2,
Yay! What do you think this region of the DNA is up here? The enhancer. So there's other transcription factors that bond to that enhancer, right? And so because of the way that DNA can loop around, those proteins attached to the enhancer are going to also interact here.
2, 1, go.
There it goes.
The blue molecule racing along the DNA. It's unzipping the double helix and copying one of the 2 strands.
Okay, so again, this is RNA polymerase, right? It's unzipping the molecule, adding the RNA. Yeah, it goes
The
back.
chain sticking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's.
Hey, I do think this is valuable to listen to, so actually listen to it.
Gene. The only difference is that in the RNA copy, the letter T.
What do we replace T with in our RNA? No, we wouldn't care. T with A, but we're replacing U. There we go.
Is replaced with a closely related building block known as U. You are watching this process called transcription in real time.
That's as fast as it
It's
like
happening
it's
right
going
now in
out
almost
fast.
every cell in your body.
Okay. So it did not. It also did not show the splicing happening. So that's simplified.
I want to see the Pac-Man.
All right, so I got some questions for you to talk about in your groups now. So here's the first question. What happens during the splicing process? All right. So here says RNA splicing. So here are the introns, the lighter pink are getting cut out and exons are getting put together. There's actually something interesting here. So because we have these intron segments, we can either splice out one exon along with it or the other exon. Okay. So that's called alternative splicing. Can create different proteins with the same gene, the same genetic code. Talk about in groups. How does that happen? Why does it create different proteins?
It's still DNA.
Then you die. Then you explode.
Okay. So. So basically, and one of the advantages of doing this process, so prokaryotes don't do, this is only a eukaryotic thing, right? But part of the advantage of this might be that when we do the splicing process, we can put certain segments of the gene in and keep certain segments out. Okay, so you're basically making different codes for different proteins using the same DNA, right? So if we only keep certain segments and pick other segments out, we're going to end up with a different mRNA code, which is eventually going to lead to a different protein, right? So that might be some of the advantage to having these introns here, right? So that we can actually create different proteins using the same genetic code. So take a close look at that. What do you think the red and the green sequences below are supposed to be representing?
Oh yeah. Are you? There's a lot more
Okay, hold up a second. So, what do we have here on the first line? What is this supposed to be?
DNA.
Is it DNA?
RNA.
Okay. How do you know it's RNA?
U.
Yeah, it's got uracil on it. Okay. So on the second line, we have the same strand, but what's missing?
Red.
All the red bases. So what are the green? Exons. What's the red? Introns. So we have our exons, we have our introns. Here's the next question.
Yes, it is.
I was just going to say you all right. And you could just like, this is so bad. Kick yourself in eyewash. Kidding. I cannot.
All right. What's this supposed to be showing here, do you think? We haven't talked about this yet, but what are these brown things?
Probably not.
Yes,
Not how would you, like, what are the words, what are they representing?
Gene. Thing.
What is the mRNA eventually going to code for?
Protein.
Protein. These are amino acids, okay?
Are you sure they're not?
So, this sequence of mRNA would code for this sequence of amino acids. Okay? If we don't do the splicing correctly, so notice one of the intron bases got left in, how does that affect your amino acid sequence? Yeah, it changes it, right? And so, if this is the protein you're supposed to have, you no longer are getting that protein, right? So, potentially this version's not going to work. Most likely, this version's not going to work. Okay. So, if we mess up the splicing, you're going to have the wrong code. You're not going to be able to make the protein properly. Okay. thought you guys were all gonna be like dead and tired today, but apparently not. Questions on this before we move on? Questions before we move on? Feel pretty good about splicing? Okay.
Got you.
So I did want to give you some time to get to the quick check. So when you look at this, first of all, on the 5' end, there's what we call the 5' cap. And on the other end, we have the poly A tail, which is, this says like 150 to 250 A's. Okay. This is really important. There's a lot of talking, and like you said, this is really important. We need to protect the mRNA from degrading enzymes. So again, one of the ways that we protect ourselves from not making too many proteins at the same time is that these mRNA molecules are eventually going to get broken down. Okay, so we don't keep making the protein when we don't need to. But we don't want it to get broken down right away. So that 5' cap and that poly A tail are going to help protect our mRNA molecule until we can get it coated into a protein. Okay, so that 5' cap and that poly A tail are going to be important for our mRNA molecule. Also, of course, we cut out all the introns, right? Again, so we've done that splicing process. Even then, we are only going to use a certain portion of that mRNA molecule to code for a protein. So there's extra bases here, there's extra bases here. So we'll talk about how we know where to start that protein tomorrow. Well, maybe not till Wednesday, but eventually we'll talk about how we figure that out. So that's the lecture. You can go ahead and get your Chromebooks out, take a look at the quick check.
Or so I think